Okay, let me get to my point. So I want to start off by talking a bit about enhancers. And the basic model for action of an enhancer, and this has been around for about, I don't know, 25 years or so, is that you have a sequence, as in, for example, in an exon here, which recruits an SR protein, and this will stimulate the binding of some sort of constitutive splicing factor at the 3' or 5' splice site. And the underlying model that people didn't really actually formalize for many years was that these encounters are mediated by three-dimensional diffusion of a freely jointed chain. In other words, the RNA is flexible and the proteins just met each other through collisions. And this was actually given a formal description in a paper by Gravely and Hertel and Magnatis, where they tested this model by increasing the distance between an enhancer like so and a 3' splice site, and they argued that their results supported that model. Now, actually, it turns out if you take their results out of the paper and reanalyze them, it turns out they don't support the model. But the other piece of evidence, which I think is a fantastic set of work, was done by Shen and Green in particular, but I mean, I suppose also by Juan, now I think about it, U2F65. And this is looking at cross-linking between proteins bound to enhancers and strategic sites, like the branch site. And this again appeared to indicate that direct contacts could be made between different parts of the RNA through space. And so far, so good. But you might want to ask yourself, well, actually, how good? I mean, is there anything that could be wrong with this? I mean, apart from the sort of flaws in the experiments. And the first thing that's wrong with it is that these lines of experiments have all used tethered RS domains. They've not, in fact, been done with intact SR proteins for good technical reasons. And the question is, does a tethered MS2 RS protein fusion properly reflect the action of an SR protein? The answer, of course, is no, because as Adrian and others have shown, but principally Adrian, in fact, the RS domain is simply not needed in the case of SRSF1 for many of its functions in splicing, constitutive splicing and alternative splicing. So how good a model is it where your system is based upon an RS domain, which actually isn't really perhaps the important bit anyway? And we've attempted to address this in one or two ways recently. Indirectly, we did some work where we showed through single molecule methods that we had U1 SNRPs able to bind to alternative 5' splice sites. Both splice sites are occupied at the same time. And if we apply a sort of random diffusional model for the RNA chain, then we'd expect to see that as we increase the distance between the alternative 5' splice sites here and here, then we'd see a shift towards the nearby site here, rising like this with distance between the sites. But in fact, what we see is a very dramatic increase like so. And this is clearly inconsistent then with the diffusional model. And we came up with some other suggestions to do with the rigidity of the exon, which are wholly speculative and probably will be rejected in their own due course. But a more direct test was one that we published a little while ago in Angavanti, where we took a leaf out of Mariano's book. Mariano, give us a wave. I can't see you. I'm a bit short-sighted. Oh, fine, thank you. And this is quite simple. Again, two 5' splice sites. There's an exonic splicing enhancer. Drag splicing over to this side. Now, let's interrupt the RNA chain by putting in something different, something plastic, like polyethylene glycol, in here, and you lose the action of the enhancer. So that says that we're not seeing interactions by free diffusion between a protein bound to the enhancer and the nearby 5' splice site. That simply isn't going to be the mechanism, unless, of course, there's an artifactual explanation, and I'm sure there are many possible explanations. Okay. If you're going to look at an enhancer and the way in which it works, then what you would quite like to use is a small defined sequence that you can isolate from the rest of the chain, where you can study what really is binding to this enhancer element to start with, and then try to follow what it does to the RNA chain when it binds. And of course, a very good model is one that we provided some years ago, more years ago than I care to think, with this oligonucleotide here. So this is SMN2 exon 7, which Adrian was talking about earlier, and this is our attempt to stimulate splicing of this exon. Here's an oligonucleotide that is composed of two domains. It anneals along here to the exon, and then there's a tail to which we hope SR proteins will bind. And here it is blown up a bit. We call the oligo GGA because the tail sequence here consists of a whole load of GGA repeats. There are 6 GGA motifs in there. This is the bit that anneals to the RNA. And there were several striking things about this oligonucleotide. We designed it to bind SRSF1. And as the years passed, binding motifs, optimal binding motifs for SRSF1 fluctuated from paper to paper. But I think actually, which worried us greatly, but I think now the consensus is from the structural work and from the clip and so on, that actually, and from the RNA compete experiments, that GGA is a good starting base if you're trying to recruit SRSF1. So we've heaved a sigh of relief there. Okay, so this we suppose recruits SRSF1. No other sequence that we've tested works very well. Even though there is a TRA2 binding site here in the exon, if we try to recruit TRA2, has almost no effect. So that particular tail sequence is particularly effective. Another point that's important is that it should anneal across this part of the exon. We've tried annealing to different positions, and in some cases it's completely inactive. In other cases, it's just not quite as active. This is the best position by some margin. The other point about the annealing domain, which is quite a strange one actually, is that if we replace these nucleotides with LNA 2' methyl nucleotides to stabilize the interaction with the RNA, to tighten the binding, we actually find that we lose activity. So it is actually apparently important for this oligonucleotide that it is not bound stably. Yeah, you explain it to me, I don't know. But this is just an observation, right? And, okay, so far so good. Did it work? Well, actually it did work very well in in vitro splicing. I'm going to show you some examples later. Worked very well in cell lines. And when Daniel Schumperle put an oligonucleotide with the same tail sequence, actually he used one that hybridized down here, but he put an oligonucleotide with the same tail sequence transgenically into mice. They were, as far as he could tell, effectively very much like normal mice. I mean, they were born, they grew, they ate, they wrote passages from Shakespeare, or whatever else it is that mice do. They did all these things pretty much just like a wild-type mouse. And that, incidentally, is why those of you that have homes that are plagued with mice, where you find a nest of mice, there's always lots of crumpled paper. It's because they've been writing their Shakespeare on it. Oh, crumbs, I said that wasn't even a joke, because I said I wouldn't. So, all right, SMN2, fine. You've got the message about the oligonucleotide. Good. Now let's find out how it works, because it's a good model system. We've been using it for years for rescuing SMN, but actually we don't really know how it works. Let's have a look. And so we tried using some hybrid fusions, hybrids with beta globin sequences, to work out which set of splicing events around the exon were affected by this oligonucleotide. And so these are in vitro splicing assays with radioactive RNA in nucleic extracts. So here we're putting exon 7 with the beta-globin exon on the left. There we have the beta-globin on the right. There we have a bit of the extra intron up here, upstream of this SMN exon 7. So that's a bit of intron 6. And these are just splicing reactions, fairly typical reactions. And the oligo makes no difference. But if we, because instead of being complete substrate, these are just partial substrates. Partial SMN2 sequences. But if now we put where the SMN2 is downstream of the beta-globin, we add in some intron sequence down here, what you see is that as you extend this intron sequence, you see a quite dramatic drop in the rate of splicing, but the oligonucleotide produces a reasonably good enhancement of the rate. So you see quite a good protective effect of the oligonucleotide against the effects of the intron down here. And as a control, we use the oligonucleotide, which we call AD, which is simply containing the part of the oligo that anneals only. It's only annealing. And you'll see that has a small, but really relatively very small effect on the efficiency of splicing. Okay, so what we're looking at then is, it would appear, some way in which the oligonucleotide, if it anneals to this exon, can counteract some detrimental effect of these sequences on events involving this splice here. And obviously we needed to look at U2F65 then, which is a likely target for the action of the oligo one way or another. And what we showed is that the oligonucleotide stimulates the binding, this is in HeLa extracts, this is in 293T extracts, stimulates the binding of U2F65 to the pre-mRNA. And those are some immunoprecipitations. Just coming over here, I just want to point out that in this case, this is looking at total cross-linking to the pre-mRNA, that the annealing domain of the oligonucleotide alone is actually also able to stimulate the binding of U2F65, not as well as the intact oligo, the complete oligo, but still it's significant. But then if we look at recruitment of a more extensive complex around this substrate here, so an exon-defined complex, we can see that without the oligonucleotide, assembly is extremely poor and the RNA appears to be instantly degraded. If we include the GGA oligo, we get very robust formation of these complexes. The annealing domain alone, this short portion of the oligonucleotide that anneals, makes a sort of a partial stab at it. And we went on to show in obvious sorts of ways by depleting U2 and by serolin cross-linking and by looking at spliceosome assembly as a whole and looking at the formation of complex A, that this involves the recruitment of U2. So U2 SNRPs are recruited by the GGA oligo, which all starts to make sense. Now, if the oligonucleotide does this, then the question, of course, is what actually is the oligonucleotide binding? As I said, we designed it to bind SRSF1. And when in about 2003, a visiting Russian did some affinity purification on the oligonucleotide and got gels looking rather like this, I was deeply skeptical, disappointed, in fact, because I had expected a single protein band. How very naive of me. But you know, he had these really clean affinity purifications, hardly any background binding to the matrix, the beads or random RNA bits, and still he got this very substantial ladder of bands. And I thought this is one of the normal problems that people encounter when they do affinity purification. Most people, when they do affinity purifications, use their RNA at too high a concentration. So they probably titrate out their limiting factors and they bind lots of secondary proteins because there's no competition for binding to the RNA. So I thought this is just the simple explanation, but actually repeat these experiments as we might, we couldn't really improve the situation beyond this sort of pattern here. And there are four major regions. This region of the high molecular weight proteins appeared to contain DHX36 and nucleolin. There was a cluster of H and R and PF and H proteins here. This band, which by mass spec appeared to contain A1 and SRSF1, which is a bit of a relief, and this, which was CNBP. So we tried another strategy where we put a photocleavable linker in between the annealing domain and the tail. And the idea is that having bound whatever complexes might form, we could use UV light just to release the ones that were attached to the tail. And we did this and we did the mass spec, and we came up actually with a fairly similar set of answers, but enabled us to say with a moderate level of confidence that actually the important proteins here, in this case the DH36, they're nucleolin and so on, as I've indicated to you. So actually it simply reinforced the original work. So we're going to have to live with this idea that there are these proteins, this cluster of proteins, this quite considerable range of proteins that are bound to the oligonucleotide. And this is just an example of cross-linking to the tail. And we've done this cross-linking by labeling the tail oligo, and we're not now digesting the ribonuclease as you do in normal cross-linking experiments. We just run the complete mixture on the gel, and you can see then that there are a number of bands that appear, and so these will be the equivalent of these bands up here. These are the H and R and PF and H bands. That's the SRSF1, and this is CNBP. If we overexpress CNBP, you can see a bit of an increase in that one. If we overexpress SRSF1, then you can see that we get these very heavy dominant bands, which are GFP SRSF1 there. Interestingly, if we immunoprecipitate with anti-H and RMP, we bring down just H and RMP F and H. So the H and RMP F and H, in most of its complexes on the oligonucleotide, is the only protein occupying the oligonucleotide. The other proteins that cross-link to the oligo are not present at the same time as H and R and P, F and H in the majority of its complexes. Because we don't see, if we immunoprecipitate with F and H, we don't see these proteins down here or up there. But if we wrap up. All right, okay, fine. If we do it with SRSF1, then we see that these proteins come down. Let me run on quickly. We asked ourselves why we've got nucleolin there, and nucleolin, DHX36, and CNBP are associated with quadruplexes. So actually, we looked at the purified oligonucleotides. Turns out that they do form quadruplexes, and we can show by reverse transcription that they appear to form them in the presence of nuclear extract as well. And this is the type of quadruplex they form. It's actually a dimer. And the question then is whether this contributes to the activity of the oligos. And it turns out that if we use quadruplex stabilizers, we compromise the activity of the oligonucleotides. And these are some in vitro splicing assays with increasing concentrations of the quadruplex stabilizers. And if that's the inclusion and that's the exclusion product, you can see it as you increase the concentration of the quadruplex stabilizer, you see a shift towards the skip form rather than the inclusion form. So it does look as if the quadruplexes are certainly not enhancing the effect of the oligonucleotides, they're probably antagonizing it. So we appear to have these three complexes that form. There are the quadruplexes associated with these proteins here, SRSF1 and H and RNP F and H. And I've shown no overlap there because Fred Alain's group and others have shown that the binding of H and RNP F and H is in competition with quadruplex formation. We've done some knockdowns to show that H and RNP F and H have no effect either on the splicing efficiency. So it looks as though the major complexes formed on this oligonucleotide, that's to say the quadruplex and the associated proteins, and the H and RNP F and H complexes are not contributing to the activity of the oligonucleotide. And these are the splicing assays, which I don't have time to tell you about, but they're very nice splicing assays. Take my word for it, they're very beautiful. So what then do we think are the mechanisms underlying this? So I want to draw here on some work from Juan and Adrian a few years ago in this paper here, where they argued that in SMN1 you had important roles for an SR protein bound here, SRSF1, U2AF, and TRA2. And the problem for SMN2 was that whereas the contribution then of this SR protein to recruiting U2AF was eliminated by the absence of this SR protein because A1 was perhaps replacing it, then you weren't going through the first stage, you weren't going through the initial recruitment of U2AF. U2 was therefore not bound, but U2, they argued, was probably dependent upon this TRA2, which would be bound here. So in SMN2, you can actually, you've got the TRA2, it's not enough on its own though for recruitment of the U2 SNP. In our case, what we suggest is that with this oligonucleotide, when it anneals, then we can now recruit U2F, but U2 binding is very weak, but with the full tail, then they have these heterogeneous complexes, but the SRSF1 is able to, together with U2F, to recruit U2. TRA2, by the way, is completely dispensable, I haven't shown you that, completely dispensable in these reactions. So this partly answers one of our questions. One of the questions was, if we use this oligonucleotide therapeutically, why would this tail not bind the endogenous SRSF1 and create problems for all sorts of other splicing reactions? Why wouldn't it sequester the SRSF1? And I think the answer is actually because the free oligonucleotide forms these complexes. And that's the reason why it isn't titrating out SRSF1, but that it's only when it's annealed and forming contacts, perhaps, that it recruits the SRSF1. That's our model. We've tried very hard to establish whether or not there's an increase in SRSF1 binding to the oligo when it's annealed to the RNA. But as I've just explained to you, the oligonucleotide is only effective when it binds very weakly, so that's been quite difficult to establish. But there's no doubt that SRSF1 binding levels do increase when the oligonucleotide anneals to the RNA. So we assume that probably this is a state then that is favored for some reason once it's bound. Where does this take us? By the looks of it, to a complete dead end. Thank you, Jamal. Thank you very much. Sorry. Mark Searle at Nottingham, who's a chemist, was was in an audience in a talk given by my collaborator throughout all this work, Glenn Burley, who's a chemist at Strathclyde. Mark Searle is the one who picked up the possible formation of the quadruplex and proved it, and Lawrence Hurley in Tucson provided the quadruplex stabilizers.